Guys, this is Mrs. Foy, and this is a screencast on the introduction to animals and a little bit about their evolution. So if you are watching the screencast, you're probably in our class, my class, Miss Dyer's and my class at York Suburban High School, and you have access to this presentation. If you are not in our class and you would like access to this, please let me know and I will give it to you. So this is just a whirlwind tour of the animal kingdom to learn a little bit more about our kingdom. And of course, we are in the domain Eukaryae. So what are the characteristics that set animals apart? Well, animals are multicellular. They are heterotrophic eukaryotes with tissues that develop from embryonic layers. And we have a wide variety of animals when we think about them all over the world. But I want to take a little step back and talk about what these embryonic layers mean and where tissues come from. So remember that almost all animals, as I said, are multicellular. They are eukaryotic organisms, so they have to, they don't make their own food. They have a nucleus. They lack cell walls. And because they are multicellular, their cells have to be held together. And there are some structural proteins, such as collagen, that do that. And there are actually 4 different types of tissue in animals, which we'll find out a little bit later on in the unit. There is epithelial tissue, connective tissue, nervous tissue, and muscular tissue. And so we are finding that nervous tissue and muscular tissue are actually unique to animals. So in order to understand where tissues come from, remember tissues are a bunch of cells that work together for the same function. We need to go back in time to a zygote, right? A fertilized egg. And we look at what happens to that zygote. And basically what happens is as soon as the sperm and the egg meet, that forms a zygote. And that zygote is a 2N organism that goes through mitosis. And what we see in this particular type of mitosis, it's called cleavage. And cleavage means that the cells are going through mitosis, but they're not getting any bigger. They're not getting any bigger. And so they continue to go through cleavage until they form a hollow ball of cells called a blastula. So notice that these are multiple cells. Now in a human, this would still be happening in the fallopian tube. This is where fertilization occurs. And so this blastula is kind of tumbling down through the fallopian tubes. The little fimbriae are moving it, the cilia are moving it down. And if you looked at the blastula, you would see that it was hollow and there's a blastocele inside of that. The next stage of development that occurs after cleavage is something called gastrulation, and it involves this amazing migration of cells. And what happens is at one end of this blastula, there is an invagination. So you can think if this was like a ball of clay, and there is an invagination, like if you stuck your thumb through that hollow ball, and the cells begin to migrate in, and they form something called an archenteron. An archenteron is the name we give for a primitive gut. So this is the very primitive digestive system that is beginning to form here. And so when we have this invagination, we can see that we form 2 layers of tissue. We form an ectoderm, which is tissue outside, and we also form an endoderm. And so the ectoderm and endoderm are tissues, they're called germ layers that form. There is also another in higher animals called mesoderm, which we'll talk about in a minute. The actual whole is called a blastopore. So keep in mind, because a little bit later on we'll talk about the 2 very different destinies of that blastopore. So later on in development, after gastrulation, the next phase of development is called nurulation. And what happens in the ectoderm, this is going to become nervous tissue. There is a very strong supportive rod of tissue that forms along the embryo, and that's called the notochord. And the notochord has its roots in what will become the vertebral column in higher animals. The tissue that lies over it is induced, so this notochord induces or kind of bosses this tissue to begin to fold up and fold a little tube, and we call this the neural crust, and it forms a long hollow tube that goes all the way up and down the length of the organism. This neural tube is going to enlarge at one end and become the brain, and then the other end is going to become the spinal cord. So just to kind of give you some background there. So I am going to ask you if you have access to this presentation that you watch this video, and hopefully this will come out okay. Here, I'm going to try to talk over it a little bit. This is actually a frog development. So here we have a frog zygote, and we can see cleavage occurring here. So this is mitosis, and you can see the cells are dividing, but they're not getting any bigger. And so cleavage continues. Now this is a little bit different from a human because amphibians actually have kind of a yolk sac that you will see that because they don't have a placenta. So it makes it a little bit different. But what's happening now is that we are having, there we go, there's invagination. So this is the beginning of gastrulation. And inside, 3 different germ layers are forming. Now on the outside, you can see we're going to see the beginning of nurulation, and you're going to see almost like a ditch form, and that is the neural tube. It's going to fold over. One end, this end up here, is going to become larger, and that's going to become the brain. And then this tube down here is going to be continuation of the brain tissue or that nervous tissue, and that's going to become the spinal cord. And then we see further development occurring. And in just a minute, you're going to see segmentation, and that's where you see little body segments. But now we begin to see the beginnings of an embryo. So hopefully you can take a class in development because it is really, really fascinating to learn more about this. So here we have our little tadpoles on their way to being developed. So animals have 3 what we call germ layers. These are embryonic tissues, and this is after gastrulation. So we have ectoderm is the germ layer covering on the outside of the embryo. Endoderm is the innermost germ layer, and that is going to line this digestive tube that we call the arc enteron, which is the primitive gut. Diploblastic, diplo means 2. Diploblastic animals are more primitive, and they only have 2 tissues. They have ectoderm and endoderm after gastrulation. But triploblastic animals, which we are a part of, are more advanced animals that have a mesoderm layer in between the endoderm and the ectoderm. And this includes all bilaterans. So these are animals that have bilateral symmetry. In other words, they have a head. And we'll talk about that in just a minute. So this is the outcome of what these different tissue layers, these germ layers, develop into. So ectoderm develops into our epidermis and nervous tissue and some other things that you can see here. Epithelial tissue is tissue that lines any opening in the body. Endoderm is going to develop into the digestive tract, the respiratory tract, the lining of certain tissues, and some of our accessory digestive organs. And notice these are the linings of the digestive system and the respiratory system. The mesoderm is going to become our bones and our muscles and the excretory, circulatory, and reproductive systems. So that's kind of the outcome of where these germ tissues are going to develop into. So most triploblastic animals with the mesoderm, ectoderm, and endoderm possess a body cavity. And a true body cavity is called a coelom. And animals that have a true body cavity are called coelomates. So when you, hopefully you've done some dissection, when you dissect and open up the abdominal cavity, say of a cat or a fetal pig, you can see that it really is a hollow cavity inside. Yes, there's organs in there, but it's not a solid thing. And that's what a body coelom is. So a true coelomate, here we have an annelid, which is an example of what an earthworm is, and we can see the true definition of a body coelom. So we can see that this is the blue is the ectoderm, the yellow is the endoderm, and the red is the mesoderm. And we can see that there is this cavity, this little space. Now look, I'm looking at a cross section here. This cavity here is going to be the body coelom, and this is where the organs are going to develop. Notice that it is completely surrounded by mesoderm tissue. That is the definition of a true body coelom. Now, this is a worm that might look a little bit like the other worm that I just showed you. It doesn't, this has segmentation, which is a more advanced characteristic. This is a pseudocoelomate. This would be a worm like a nematode worm that we call a pseudocoelom. So let me explain why. It's a very subtle difference. So notice that it is triploblastic. I see an ectoderm, I see an endoderm tissue. This is going to be my digestive system. Notice it's a tube. And I do see mesoderm, and I do see a body coelom. But notice that it is not completely surrounded by mesoderm. Okay, let me go back here and show you this. Do you see how this body coelom is completely surrounded by mesoderm? And this one is not. And so this is a less developed, this is a less advanced animal. Now we go to this other kind of worm that we would call a flatworm, and this is an acoelomate animal. It is triploblastic, but it has no body coelom at all. And so we would not expect to see developed organs in this particular animal, which we in fact do not, with the exception of sexual development organs, sex organs, which we do see. Now, do you remember talking about the blastopore? Remember that invagination that happens in the blastula that starts gastrulation? Well, there is a very different destiny of that blastopore in two major kinds of animals. The first are what we call the protostomes. These are the mollusks, any kind of shelled animals, and annelids like we saw the segmented worm, the earthworm. In these organisms, the blastopore becomes the mouth. And the archenteron, as it forms, the secondary hole in the tube becomes the anus. So mouth forms first and then anus. This is in a protostome. This literally means first mouth. The other major kinds of organisms form, they are called deuterostome formation. And the blastopore in a deuterostome actually forms the anus first, and then secondarily the mouth forms. And so deuterostome, deutero means second, so this means second mouth. And so the anus forms first and then the mouth. And you can see that examples are echinoderms, which are like starfish. Guess what? Vertebrates, like us, are also deuterostomes. So believe it or not, we actually form anus first. Anus and then mouth for deuterostomes. I want to talk a little bit about body plans in animals. The most primitive type of body plan is asymmetrical. So sponges are the most primitive animal forms, some of the most primitive animal forms that we have. Radial symmetry is what this cnidarian or the jellyfish family are showing. And bilateral symmetry is when you have mirror images on the left and right. Bilaterally symmetric animals are usually the higher animals that we think of, and they have a head. And the fancy name for that is called cephalization, and that means that you have a head. If you have a head, then you usually are bilaterally symmetrical. So, hold on a second. Sorry, my lights are timed to go off. So, anyway, this is a family. This is a family tree of the animals, and let's see if we can figure out what these things are. So we have the porifera, which are the sponges, the most simple organisms that are in the animal kingdom. One of the characteristics of animals is that they are mobile, they move, and the particular, you might think, well, a sponge doesn't move, it just sits, it's sessile, right? It doesn't move. But the larva form of a porifera does move, and so technically that's why we stick it in this kingdom. So we have the ancestral Protista, which are one-celled organisms that we think were the ancestors of animals, which we'll talk about in a minute. And these are asymmetrical, remember. And then we have our radial ancestors, our radial symmetrical ancestors. And now we have the diploblastic organisms. So like jellyfish, the cnidarians, they do not have a mesoderm, but they have endoderm and ectoderm, and they are radially symmetrical. And then we have our ancestral triploblastic acoelomate. So triploblastic, remember, means that there are 3 germ layers: ectoderm, endoderm, and mesoderm, but they are acoelomate, so they have no body coelom. So here are flatworms like our platyhelminthes that are a lot of, there are some parasitic organisms that are in this category. Our pseudocoelomates, remember, these have a false coelom, so they do have a body coelom, but it's not completely surrounded by mesoderm, and these would be like nematode worms. Some of these are important parasites as well. And then we have our coelomate organisms. So this is a true body coelom, completely surrounded by mesoderm, a body cavity. So now we're expecting to see organs in this body cavity. And then we have the 2 great divisions of the animal kingdom. We have the protostome lineage, so the mollusks, the arthropods, these would include the insects as well, and the annelids all form mouth first, so that blastopore becomes the mouth, and then the anus forms secondarily. And then the deuterostome lineage are the chordates. So most chordates are vertebrates. So these are animals with backbones, of which we would belong, and the echinoderms, the starfish, and the sand dollar organisms, which we are also deuterostomes like them. Now, just to give you a little bit idea about when animals develop, let's look at the whole geographic timeline, right? So this is about 4.6 billion years that we have on Earth. And remember, about halfway through this time is where we very first get a cell with a nucleus. Over in the Archean era, we just have bacteria. So now we have cells with a nucleus, and we have organisms that are beginning to photosynthesize now, and so we have a rise in atmospheric oxygen. But nothing really exciting happens in these shallow oceans until about a half a billion years or 500 million years ago from today. And this is where we have the Cambrian explosion, and that's where we begin to see the evolution of animals. So in this, now I'm just looking at this time period. You see, this is not to scale, right? So this is 3,000 million, which is 3 billion. I'm just looking at 500 million, or about a half a billion years ago. And here's my Cambrian explosion, right? This is where we think that there was enough oxygen in the atmosphere to really sustain multicellular organisms like animals. So we have organisms like trilobites. These are the different periods that we have here. These are our shelled animals. These are arthropods. Devonians. You should know that Devonian is the age of fishes. And we had fishes first that had a cartilaginous skeleton, like sharks and rays, and then we developed bone or osteo backbones. In the Pennsylvanian and the Mississippian periods, we actually had amphibians. So now we have our first organisms that crawl out onto the land, and they have a dual life. Amphibian means dual life. So they still have to go back to the water to reproduce, but they can live on land and they can breathe air. And from that, we had the evolution of reptiles. And the ancestral reptiles basically split into what would become birds and mammals, and what would become, what would become mammals, and then what would become birds, and the current reptiles that we have now. So we've talked about this already in class. Remember that dinosaurs, the dinosaur period, dinosaurs were dominant on Earth way longer than mammals have been around, and certainly a lot longer than humans have been around. And of course, we know that the KT extinction event is what laid the groundwork for the explosion of mammals in the last period of geologic history. So we believe that our modern ancestors, that the ancestors of animals were choanoflagellates, and I'll show you what those look like. And this was happening around about the Cambrian explosion. So choanoflagellates, you can see that these are colonial organisms. They're little one-celled organisms that have a little flagella, and they move that flagella. They're filter feeders, and this is very similar to the way that a sponge eats. And remember, a sponge is our most primitive animal that we have in our same kingdom. And then all of the others developed from that. The Cambrian explosion about 500 million years ago, there are three things that are significant about that. We think that these were the first predator-prey relationships. In order to be a predator, you need to have good sense organs, and so this was the beginning of having a head, cephalization. So you have all your sense organs in your head, and the development of jaws. Okay, the actual development of jaws, which not all animals have jaws. And this coincides with the rise in atmospheric oxygen and the evolution of the Hox gene complex. And the Hox genes, we know, are genes that are the architecture of a body plan. So this would be a typical Cambrian explosion seascape, shallow seas, and you can see these are not imaginary. We have fossil evidence of all these crazy looking animals in the Cambrian explosion. The Hox genes that we know are the set of genes that are the master architect for a body plan, and they are very, very similar in organisms like a fruit fly on up to a mouse on up to a human. So we have this series of genes that are helping to develop a body plan. We know that about 460 million years ago, animals began to make an impact on land. So we had actually insects were first, but vertebrates made the transition to land about 360 million years ago. And this is a artist's rendition of a very important fossil find that may have shown us the very first evolutionary step toward amphibians. So there's an awesome book by Neil Shubin called Your Inner Fish. There's also a great video that he does through the HHMI website, so you should definitely check that out. This Tikalik is the fossil that he found, and it really is an important link between the evolution of more aquatic organisms and land organisms. You can see this is pretty much going from more primitive to more advanced. So sharks have a backbone. They are vertebrates, but their backbone is cartilaginous. Ray fin fishes have a bony skeleton, but they are different than what we call lobe-finned fishes. And lobe-finned fishes are the coelacanth that, remember, we thought were extinct, but we found some about 50, 60 years ago. And the lungfish, which also are lobe-finned, we know that these can survive for a short period of time on land. And notice that this is not a linear thing. This is just showing a progression. So we have amphibians. These would be ancestral reptiles that can live without going back to the water to reproduce, and now we have the rise of mammals, lizards and their relatives, which would include birds. So the Mesozoic era is when the first mammals emerged, and these were rodent-like organisms, and of course the dinosaurs were very dominant during this time of Mesozoic era. Coral reefs, we begin to see the development of coral reefs as well. The Cenozoic era was the era that followed after the extinction of dinosaurs, that we have very strong evidence that was the KT event, that was this huge asteroid that hit the Earth and caused the extinction of dinosaurs. And notice that it says non-flying because those other flying dinosaurs, we know that they evolved into birds. And the mammals and insects really diversified through the Cenozoic. So I want to talk a little bit about the evolution and how we classify these organisms. So an amniotic organism is an organism that develops from an egg. And so this was the first time that animals were not tied to the water. So you think about a fish egg and how it was very jelly-like, and then you think about like a turtle egg or a bird egg, that there is actually water, the amniotic sac that surrounds that, but it is in a waterproof egg, so to speak, that allows that development. So we had a particular type of organisms called the synapsids, and then finally the therapsids, that were more mammal-like. And these are going to develop into, you probably recognize this as a warm-blooded, that we can control our own body temperatures. Lactation means we begin to feed our young milk. And the mammals are actually have developed into three main groups. The monotremes, which are the very weird mammals. They actually have fur, they feed their young milk, but they actually are egg-laying, and this would be like a duck-billed platypus. Only see this in Australia. And the marsupials, which we also mostly see in Australia, these are the pouched organisms, mammals, that have prolonged lactation. So the babies are born very, very prematurely, but then they develop into the pouch. They kind of latch on and begin to suckle milk, and we know that pandas are like this as well, and they just develop longer periods of time, but these are the marsupials. Finally, we have the eutherians. These are the placental mammals, and so these organisms do not have a pouch, and most mammals that we think of are eutherians, have a placenta. So this is just showing you a phylogenetic tree of these organisms. We have the ancestral protists, we have the development of tissues, which would be on multicellular organisms. We have bilateral symmetry. We get the beginnings of a body cavity. We have two different major types of organisms. We have a deuterostomes and our protostomes. Some of these protostomes are actually pseudocoelomate, and these guys do have coeloms. But these are where the chordatas are. And so you can just kind of take a look at where the different groups of animals, we actually have these are more traditional names for these animal groups, and if you take another class in animal classification and anatomy and physiology, you'll probably learn some different classification names that we have for these today. So I hope this has been helpful to you, and I will see you in class.